Hello student, welcome to EPG Pathshala. I am Dr. Vishwanath Tiwari from Central University of Rajasthan. Today we are going to talk on the module that is enzyme regulation of aspartate transcarbamylase, metalloenzyme carboxypeptidase A, and the role of metals or zinc. This module has been taken from molecular enzymology and protein engineering. So, the objective of this module is to understand the regulation of different enzymes, and we are taking aspartate transcarbamylase as an example to explain enzyme regulation. We have also taken the other example of carboxypeptidase A, which is representing metalloenzyme, and we will also discuss what is the role of metals and other factor in the process of enzyme regulation. So, in this module, we will discuss enzyme regulation by taking few examples. So, what are allosteric enzyme? So, allosteric enzyme are having two sites. One is active site, another is allosteric site. So, allosteric enzyme follows two mechanisms. The first one is allosteric inhibition and second one is allosteric activation. So, let us first talk about allosteric inhibition. So, as you can see in the structure, we have one active site and other allosteric site. Allosteric inhibitors bind to the allosteric site, and binding of allosteric inhibitor to the allosteric site will result into the conformational change. As you can see in the structure, when allosteric inhibitor is present into the allosteric site, it will distort active site, and distorted active site are unable to bind with the substrate, that leads to the decreased activity of the enzyme. Therefore, you can say this is allosteric inhibition. The second class is allosteric activation. As you know that in normal circumstances, your active site is distorted. So, you need allosteric site to bind with some molecule to activate or to regain distorted active site converted into normal site. So, allosteric activator will bind to the allosteric sites, and binding of allosteric activator to allosteric site result into the conformational change in the active site, which promote its adopting a conformation which is its native functional conformation, and the substrate will bind to it. Binding of substrate to the active site is followed its conversion into the product. So, therefore, during allosteric activation, activator induces conformation change in the active site, which promote binding of substrate and its catalysis. How aspartate transcarbamylase involved in the catalysis? As you know that aspartate transcarbamylase is involved in the pyrimidine biosynthesis, and the pyrimidine biosynthesis have two precursors. One is carbamoyl phosphate, another is aspartate. So, carbamoyl phosphate combined with the aspartate in the presence of aspartate transcarbamylase to produce carbamoyl phosphate, and after formation of carbamoyl phosphate, the phosphate will be removed, and during this process the carbamoyl aspartate is produced, which subsequent cyclize and producing orotate, and which further catalyze the production of UMP, and in the subsequent reaction, this UMP is converted into TMP, and uracil and thymine that differ only in the amino group. So uracil is converted to cytosine with the help of CTP synthase, and that require glutamine, and that is converted into glutamate. So the CTP is synthesized after the pyrimidine biosynthesis. So when we are going to start with the carmyl phosphate and aspartate, that will lead to the production of UMP, that is converted into CTP, and that also produces UTP, and the UMP is converted into the TMP. So we will have production of 3 pyrimidine, cytosine, uracil and thymine by this mechanism. And here you can see that CTP, that is a final product, or you can say one of the final product of pyrimidine biosynthesis, is inhibiting aspartate transcarbamylase. At the same time, it is also inhibiting CTP synthase. So in this way you can say that CTP is feedback inhibiting aspartate transcarbamylase. It feedback inhibiting the CTP synthase, because CTP is a final product and these are the intermediate enzymes that is involved in the synthesis of CTP. In this table you can see what are the contribution of different scientists in the field of ATKs. You can see that the Purdy and Yates explain the control of biosynthesis by a feedback inhibition mechanism for pyrimidine nucleotide biosynthesis. Purdy and Sifedson explain, or you can say the purified ATKs and its crystallization. Purdy and Gerhardt studied the CTP and ATP can affect the substrate binding in indirect mechanism, or you can say it's allosteric mechanism. And Lipscomb give the X-ray crystallography structure of ATKs. So what is the quaternary structure of ATKs? As you know, quaternary structure is only formed when a protein is oligomeric in nature. And quaternary structure explain the arrangement of different subunits with respect to each other and their nature of contact. So native enzyme of ATKs is dodecamers, which consist of regulatory and catalytic subunit. So catalytic subunit is represented by C and regulatory subunit is represented by R. So catalytic subunit is large subunit. It consists of 3 chain. Therefore, we are saying C3, that is representing C for catalytic, 3 for 3 chains. So the catalytic subunit is only involved in the catalysis, but it doesn't have any effect in terms of its regulation by the CTP. While the regulatory subunit is the smaller one, it consists of 2 chain and it is denoted by R2. An interesting thing is it is regulated by CTP and ATP, but it doesn't have any catalytic activity. So overall quaternary structure of ATKs is consist of 2 catalytic subunits, that is C3 plus C3 to become C6. At the same time, 3 regulatory subunit. So 3 multiplied by R2 should become R6. So this is a native state having 2 catalytic trimer and 3 regulatory dimers. You can see in the structure how the different subunits of ATKs are arranged, where you can see that C1, C2 and C3 corresponds to 1 catalytic trimer. C4, C5 and C6 correspond to the second catalytic trimer. Similarly, you can also see that R1, R6 are 2 regulatory subunits which are present close to each other. R2 and R4 are other regulatory subunit which is present close to each other. Similarly, R3 and R5 are other catalytic subunit which is present close to each other. So in this way you can see that we have 2 catalytic trimer and 3 regulatory dimers. Here you can see that how a catalytic trimer present surrounded by a regulatory dimer, and you can also see the important domain, that is zinc domain, that is present there, and you can also see how R chain, RC chain are going to present close to each other, and what is their stoichiometry, what is their arrangement in the three-dimensional spaces. So what is the secondary structure of ATKs? Secondary structure, as you know, is consists of alpha helix, beta sheet. When you have super secondary structure, it is motifs and domain. So catalytic chain of ATKs is composed of two folding domain. The first one is spartate domain. It is primarily involved in the binding of spartate. And the second domain is carboyl phosphate domain, which is mainly involved in the binding of carboyl phosphate. And you know that this ATKs have two substrate, one is spartate, other is carboyl phosphate. So both these domains are involved in the binding of those two substrate. The second domain is regulatory chain or regulatory domain. It consists of two folding domain. The first one is zinc domain, which is primarily involved in the binding of zinc cofactors, and the allosteric domain, which is involved in the binding of allosteric effectors such as CTP, etc. You can see the location of different domain in ATKs. This ATKs is represented here in the T state, which is non-functional state. So you can see how AI domain, that is present in the regulatory chain, what is the location of zinc domain, which is present between regulatory chain and catalytic chain, and how aspartic acid domain and how carboyl phosphate domain are present in the three-dimensional spaces. So here you can see the catalytic domain, aspartic domain, carboyl phosphate domain, zinc domain, you can say it behaving like linker, and then we have allosteric regulatory domain. Let's have introduction of aspartate transcarbamylase. Aspartate transcarbamylase is also known as ATKs. It has enzyme commission number of 2.1.3.2, and it catalyzes the first committed step in the biosynthesis of pyrimidine nucleotide. Pyrimidine nucleotide such as CTP. These pyrimidine nucleotides are important component of nucleic acid. Therefore, aspartate transcarbamylase are involved in the synthesis of nucleic acid. Specifically, aspartate transcarbamylase participate in the condensation of aspartic acid and carbamoyl phosphate to form N-carbamoyl aspartate and orthophosphate. This is very crucial reaction because aspartate transcarbamylase catalyzes the second step of pyrimidine biosynthesis, and this step is important when you are talking about complete pyrimidine ring synthesis, because for the pyrimidine synthesis the most of the atom comes from the aspartate. So aspartate and carboyl phosphate condensation is important for pyrimidine biosynthesis. So after a discussion of quaternary structure and the secondary structure, now let us see what is the active site and catalytic mechanism of ATKs. So here you can see the catalytic mechanism follows an order in which carboyl phosphate bind before the aspartate and leaves before the phosphate. So this follow a bisubstrate reaction. So binding of carboyl phosphate result into various conformation change such as reorientation of AT's loop, that is serine AT and lysine AT4 into the active site. The motion of the 50s, for example threonine 53 and threonine 55 will move. The movement of 240s, that is movement of arginine 229 and glutamine 231 loops. The conformation changes after electrostatic properties of the active site and create a positive charge binding pocket for the aspartate. So these conformation changes are important for binding creation of the positively charged binding pocket for the aspartic acid. So in this figure you can see that what are the different residues that is involved in the active site of acetase. As I have discussed that the binding of carboyl phosphate always taking place before the binding of aspartic acid, so the conformational changes that is created by the binding of carboyl phosphate will basically creating the pocket in such a way that it will facilitate the binding of aspartic acid. As you know, for any allosteric enzyme, the conversion of T to R and R to T is very important. So you can see that ATKs, similar to other allosteric enzyme, is also existing in T state and R state. And binding of a pala as a molecules causes conversion of T state into the R state. And you can see that T state and conversion into R state, there is a conformation changes or there is a rearrangement of different subunit within the ATKs take place. So secondary structure of ATKs consist of domain, aspartic acid domain and CP domain. Both aspartic acid and CP domain of the catalytic chain and AI domain, that is a third domain, is a regulatory chain domain, and they are classified as alpha/beta domain. As you know that alpha/beta domain indicating that the domain consists of alpha helices as well as beta sheets. The zinc domain of the regulatory chain is classified as rubridoxin-like domain, which contain metals, zinc or iron, bound with usually contain two motifs, and that is CX in the bracket NC motif. As you know from the symmetrical model, the T state is a one that bind with the inhibitor and R state is a one that bind with the substrate or activator. And you know that CTP is an end product of pyrimidine biosynthesis. So that must feedback inhibit this enzyme. So therefore, as you can see that CTP binding is favored in the T state, therefore it will promote conversion of R state into the T state, hence shifting the equilibrium towards inactivate form of enzyme, and therefore that decreases the enzyme activity. So what is the allosteric regulation of ATKs? So ATKs follow concerted mechanism of regulation. Both ATP and CTP are allosteric effector. Effector means it may be activator, inhibitor. ATKs regulates do not follow Michaelis-Menten kinetics because this is not a Michaelis-Menten enzyme, but it exhibits the sigmoidal kinetics. It shows ATKs is an allosteric enzyme, and its cooperativity between active site promotes the ligand binding. So when we are talking about the effector molecules, the effector molecule may be activator, may be inhibitor. So ATKs is negatively regulated by CTP. That means if CTP is present, then there is no need to make it because that is an end product of the reaction. So in the presence of CTP, we need the ATKs to be inhibited. At the same time, ATKs is positively regulated by the ATP. So ATP abundance indicating that there is a sufficient energy stored in the cell which promote DNA replication and messenger RNA synthesis. So if ATP abundance, or you can say ATP is more than CTP, there is an increase in the ATK's activity which is required to balance the purine and pyrimidine biosynthesis, because ATP is a purine and CTP is a pyrimidine. In this figure, you can see that how the binding of CTP to the regulatory subunit stabilizes the T state, and it shows that the CTP binds to the T state and stabilizes the T state, hence act as an inhibitor for ATKs. So in this graph, you can see if we increase the aspartate concentration, which is a substrate of it, the rate of N-carbamyl aspartate formation is increased. And so T2R switches induced by ATP binding, because here in this reaction, we will having 2 millimolar ATP. So when we are having 2 millimolar of ATP and we are adding aspartic acid, or another word you can say, we have aspartic acid, So rate of product formation is there, but if you add 2 molar ATP, then the rate of product formation enhance, which you can see with the green color. So this shows that ATP acts as an allosteric activator, which makes the velocity curve more hyperbolic. Here you can see that we have a sigmoidal curve for if you increase the aspartic acid concentration and making a curve with reference to aspartic acid and rate of formation of N-carbonyl aspartate. But if you add CTP, then curve become more sigmoidal. It shows that CTP is behaving like allosteric inhibitor. After discussion of ATKs, now let's have discussion of another enzyme, that is a metalloenzyme. So what is the requirement of metal? So metals are required as a prosthetic group or cofactor by the enzyme, and it is important in enzyme catalysis because the free amino and carboxylic group of the amino acid can form coordinate covalent bond with metal, affecting the enzyme structure and catalysis in its active conformation. Because when you're talking about electrostatic catalysis as well as covalent catalysis, then metal is going to have influence. They predominantly help in the electron transfer because metals are electrophilic, so they resemble proton that are able to accept the electron pair to form a chemical bond. In this table you can see different enzymes and their different metal requirements. For example, hemoglobin have iron, cytochrome have iron, phosphotransperases have magnesium and it is involved in the electron transfer. Alcohol dehydrogenases have zinc, arginase have magnesium, pyridoxine have iron, cytochrome oxidase have copper, and carboxypeptidase A have zinc. So in this table you can see there are different enzymes, they require different metal. We will discuss one metalloenzyme, and that is carboxypeptidase A in this module, that represents a model for metalloenzyme. How a metalloenzyme is going to be regulated? So pH have effect on metalloenzyme because electrophilic ability of metal is pH dependent. So change in the pH may disturb the electron flow that a metal would normally help to facilitate. Therefore, by changing the pH, the enzyme efficiency of a metalloenzyme may alter. The transition state analogs may also alter the metalloenzyme activity because they act as a competitive inhibitor. So transition state analog or substrate analog, they are mimicking the active site binding substrate or transition state, therefore they are competitive inhibitors. The presence of zinc in the diet is well known. So zinc containing metalloenzyme can also be regulated by the diet. So the deficiency of the zinc in the diet can inhibit the enzyme activity because zinc is required to maintaining the structure of as well as catalysis of the metalloenzyme which is requiring the zinc. So we have an enzyme that represents the metalloenzyme, and that is carboxypeptidase A. It has an EC number of 3.4.17.1, and this is pancreatic metalloexopeptidase. It hydrolyzes peptide bond adjacent to the C-terminal end of polypeptide chain and is specific for large hydrophobic side chain. It also hydrolyzes ester with similar configuration and even with the faster rate. And it belongs to monozinc proteases or zinc hydrolases family and considered as a prototypical model for the study of mechanism of action of metalloenzyme. So allosteric regulation of ATKs is explained by symmetrical model where you know that we have two states, one is T state, other is R state. So T state is known as tense state, which has lower affinity for the substrate, lower catalytic activity, and predominate in the absence of the substrate. And the second state is relaxed RR state, which is prevalent when we have substrate or its analog, which is bound to the ATKs, and it is having high affinity for the substrate and it is having higher catalytic activity. So enzyme exists in equilibrium between T and R state at any fixed concentration of aspartate and carboyl phosphate. So the binding of these substrate to the R state promoting conversion of T into R state and therefore activating the process of catalysis and therefore activating the allosteric regulation of ATPase. So what is the structure of active site and metalloenzyme like carboxypeptidase A? Carboxypeptidase A is single chain, consists of 307 amino acid. It has single zinc in the active site. So zinc behaving like cofactor and it is stabilizes within active site by forming interaction with histidine 69, glutamic acid 72 and histidine 196, and it additionally bound by catalytic water molecule that interacts with glutamic acid 270. So arginine 127, tyrosine 248, arginine 71, spartic acid 144, asparagine 144 and arginine 145 from an outer cell will indicate glutamic acid 270 around the active site and contribute to the catalytic function of carboxypeptidase A through stabilization of substrate molecule. Glutamic acid 270 is important for catalysis either in terms of general acid base catalysis or as in the nucleophile in the covalent catalysis. Here you can see the active site pocket and how your zinc is associated with different side chain residue. You can see zinc is associated with histidine and glutamic acid residue and how the arginine is also present in close vicinity. And you can also see the water molecule that is also present close to the zinc. So this water molecule is going to be influenced by the presence of this zinc atom in the active site. So what is the biological role of carboxypeptidase A? So carboxypeptidase A facilitate the breakdown of proteins during the metabolism. As its name suggests, it is peptidase, so it's break peptide bond and the carboxy is there, so it is breaking the peptide bond starting from the carboxyl terminals. And its commercial application is, it is a hydrolysis of choice for the proteins and it is important for production of phenylalanine free protein hydroxylates which is used for the individual with phenylketonuria. You can see how the enzyme catalysis of carboxypeptidase A take place. So water molecule which is present in the active site and close to the bound zinc metal, once the substrate bind there, the water molecule plays significant role in the catalysis. So enzyme substrate complexes form where the enzyme is water. So enzyme is having active site with zinc and water. So water is activated by the presence of zinc. So when the ES complex is formed, then substrate molecule combined with the binding residues of active site and there is a formation of transition state. And then transition state is having some rearrangement in the bonds and the second transition state is formed and finally that will leads to the production of the product during this reaction. So in this figure you can see how the different bonds changes during the conversion of the substrate into the product, or another word you can say the cleavage of peptide at the carboxylene. In the previous model you can see the role, in the previous model you have seen the role of water in the catalysis. Here you can see the role of metal in the catalysis because metal acts as a nucleophile, therefore you can see it follow nucleophilic pathway also, where zinc directly involved in the catalysis. So ES complex is formed where the zinc is involved and then transition state is of the substrate is formed and which is finally converted into the product. So product is released. You can see During the conversion of reactant into the product, different rearrangements of the bond take place. So, what is the inhibition of carboxypeptidase A? So, there are two type of inhibitor that is designed against the carboxypeptidase A. The first one is active site directed inhibitor. It is characterized by presence of terminal carboxylases and a hydrophobic side chain and zinc binding groups. For example, enantiomers of 2-benzyl 5-hydroxy 4-oxopentonic. This compound interacts with its carboxylate by forming a hydrogen bond with arginine 144, arginine 127, tyrosine 248 and its terminal hydroxy group by forming a hydrogen bond with serine 197 to inhibit carboxypeptidase A. The second mode of mechanism is transition state analog. So, inhibitor bound with the active site of the enzyme and form a pseudo transition state complex that irreversibly inhibit by the enzyme. Such example is 3-phosphonopropionic acid. There are other mechanism of carboxypeptidase A inhibition and that include UV visible radiation. So, uncompetitive inhibition of carboxypeptidase A is also take place, but the mechanism is not very clear. So, decrease in the enzyme activity is indirectly proportional to the radiation time. And the last, the role of metal in the catalysis is well known. So, the diet is also important component when you're talking about the regulation. So, zinc is a key component in the diet. If zinc is absent in the diet, then it is going to basically inhibit the catalytic activity of this metalloenzyme. So, in this way, we have learned how metalloenzyme is going to influence by different inhibitors such as active site analogs, transition state analog, and how the dietary components of the metal is also influencing the activity of the enzyme. So, what is a catalytic mechanism of it? So, there are two mechanisms that is adopted by this enzyme. The first one is general acid base catalysis. So, in general acid base catalysis, as the name suggests that in general acid base catalysis, all the active site residues as well as water present in the active site are involved in the catalysis. So, they are going to play important role in promoting water dependent pathway. The second pathway that involved is nucleophilic pathway. So, this nucleophilic pathway is a part of nucleophilic covalent pathway. So, the computational analysis suggests that promoted water pathway is the preferred pathway in the proteolysis as it has lower kinetic barrier, because if any pathway having lower kinetic barrier, it is going to be favored. Therefore, this kinetic mechanism of this involve acid base catalysis as well as covalent catalysis. So, student, let's discuss what we have learned in this module. So, we have learned how allosteric enzyme is important and how allosterism is explained by taking few examples. So, allosteric enzyme constitute an important class of enzyme whose biological activity can be controlled by a specific regulatory molecules. These regulatory molecules may be activator, may be inhibitor of enzyme activity, which binds to the allosteric site and control the enzyme active sites. The allosterically regulated enzyme do not follow Michaelis-Menten kinetics. The enzyme consists of multiple subunits, or you can say oligomers, and shows cooperativity in ligand binding and displaying sigmoidal kinetics rather than hyperbolic kinetics of Michaelis-Menten. The aspartate transcarbamylase is considered as a classical model to study allosteric regulation. The enzyme catalyzes the synthesis of N-carbamoyl aspartate, that is the first intermediate of pyrimidine biosynthesis. This enzyme consists of two separate catalytic, that is C3, and regulatory, that is R2 subunits. It is negatively regulated by CTP, that is a product of this enzyme, while positively regulated by ATP. So, allosteric transition of aspartate transcarbamylase is concerted. That means all subunits of the molecule simultaneously interconvert from low affinity T state to high affinity R state. We have also seen that metalloenzyme need metal as a cofactor and consist of an array of enzymes important for many physiological processes. Metal works as electrophile and it's added or help the catalytic process of the enzyme. pH, transition state analogs and diet are the key regulator of metalloenzymes. Carboxypeptidase A is an exopeptide that requires zinc ion as a cofactor. This pancreatic enzyme hydrolyzes peptide bond either by promoted water or by nucleophilic pathway. Dietary deficiency of zinc can negatively regulate this enzyme. So, we have understood how the enzyme regulation is important for the process of studying allosterism. Thank you.